Re-
Sounds good.
Yeah, thanks. All right. Well, sorry for the brief delay there. Welcome to the Spring 2022 CCB Skills Seminar. Today, we have Monica Arnielle joining us. She'll be talking about R's Tidyverse for data science. Monica graduated from Duke University, where she majored in biology with an emphasis in genetics, and she minored in computational biology as well, in bioinformatics. And she did 3 years in the Getz lab at the Broad Institute, where she's working on non-small cell lung cancer. And now she's really interested in evolutionary biology and population genetics. And she's a first year in the CCB program. We're so excited to have you. Before we get started really quick, I just want to note that we have lunch back there. Feel free to take some lunch. We might even have some extra, but no eating inside the room. There's a sign-up sheet back there. And we're also going to have a feedback form that we'd like everyone to fill out to give us ideas for what future seminars can cover, as well as just justify to the school that this is worth spending money on. So I think those are the major announcements. Monica, we're so excited to have you. Please take it away.
Yeah, so excited to be here, and thank you guys so much for coming. I will just jump straight into the slides. So I will present shortly some slides that I have, and then the second part is going to be jumping into tutorial/demo. So I'm Monica. I'm a first year here in Comp Bio, and first I'd like to just talk about the Tidyverse, like what packages it includes, kind of what's the philosophy behind it, then sort of go into some of the useful packages that we'll be using today, and then lastly just take you through the tutorial/demo. And so first of all, what is the Tidyverse? It's one giant suite of packages in R that make it easier to wrangle, analyze, and visualize data. So that's basically the building blocks of data science, and it comes into so many operations that I do, at least in the day-to-day. So I hope that it can be helpful to you too. The core packages include ggplot2, which you might be familiar with, as well as additional ones that don't get automatically loaded in the Tidyverse, but are also kind of associated in the whole ecosystem, like database or Google Drive integrations. And then there's also a whole ecosystem of packages that build on the Tidyverse and use tidy principles. So the Tidyverse, there are some pros and cons when you actually start Googling around and asking if people like it or not. So some of the pros is that, the command names are very intuitive. Base R uses all these symbols and $signs, whereas Tidyverse is very clear with the words what each function is doing. It encourages piping commands, which basically gives your code some sort of a flow, as opposed to nesting things infinitely and hiding them in functions. So you could argue that it makes your code a little easier to read. The cons, though, is that it's very much a dialect, so it's not really similar to base R. So you wouldn't right away know what a base R equivalent of the Tidyverse is just based on a function name. And then because of that, because these function names are very kind of plain English, very intuitive, they hide a lot of complexity. So it's great to get things done quickly, but it's not so great if you're just starting out programming, or if you maybe want to write your own R package. You wouldn't do that in packages using so many packages from the Tidyverse. And again, it's not really helpful to learning programming, and it's hard to learn if you don't know programming. So it's kind of like a again, it's great for shortcuts, not so great for getting started with R or programming. So these are the core packages that I mentioned. ggplot2 is really popular. dplyr, tidyr, reader, per, Tibble, stringer, and forcats. And then these are others that you have to load manually, but are also kind of included in the Tidyverse. And one really cool one that I like and will introduce briefly towards the end is tidy models, which you can do a lot of really data sciencey things with, but I think would be another talk by itself. So for these core packages, this is basically the differences between them. ggplot2, it relies on this kind of grammar of graphics that the Tidyverse creators really stand behind, and has like a whole philosophy like attached to it. And basically, well, I'll explain it in the next slide, but that's basically how to make really customized plotting objects. dplyr manipulates data, so filters it, subsets it, alters your columns. Tidyr is more about reshaping your tables. Reader is for reading into external files. Per is functional programming, which I won't be going too much into detail about. Tables are basically different versions of data frames, which are the data tables. Well, data tables is another thing, but data frames are one of the base R data structures that you want to know, and tables are Tidyverse's response to that. So they're a little more rigid. They don't do as many things for you, like they don't rename rows automatically, and they are very strict in their typing. So in the data types that are contained in your columns. Then there's also Stringer, which does, your pattern matching, your subsetting, etc. And then ForCats, which has a lot of tools to deal with factors, which if you've been working with R for a while, that can be a headache. So these are all the great kind of core Tidyverse packages. And so this is the what I was mentioning before. ggplot relies on this grammar of graphics. So I included the screenshot because I think it helps explain a bit like the language that ggplot uses. A graph is a set of points, and a graphic is a physical representation of a graph. And this representation is accomplished by realizing graphs with aesthetic mappings or aesthetic attributes such as size or color. And so there's a whole book about this. Basically, this system is instead of having like preset graphs that you generate, like when you go to Excel or when you go to like a specific package in Python, this is more of like a language. So it has independent components that interact with each other according to some set of rules, hence the grammar. Yes. Yeah.
Is there, I guess what I'm thinking about is there a advantage of using Tibble over something like Python where you have like pandas and Python where you can data frame more easily? Is there a reason why people choose to use a more rigid Python?
Yeah, so for people on Zoom, the question was, what is the reason to use Tibbles over some more general data frame that gives you more flexibility? Like something from Python or like even data frames in R, which are a little more flexible. And, the argument for Tibbles is that because they're so strict and kind of, they call them like lazy data frames, as in like they don't do as much stuff for you, it forces you to have more control over what data types you're putting in. And I think that can be really useful because for instance, I've found myself many times like doing like a really sloppy join and then a bunch of things are dropped or coerced into the wrong type. So the creators of the Tidyverse are basically reasoning like if you're forced ahead of time to keep your Tibbles in check, then later down the line you'll have fewer problems. So that's the argument. That being said, I like that. At the same time, is it something that most people do? I don't know. I think it's up to individual preference. You just have to be more careful with the flexible data frames. So in short, the grammar of graphics is just like language grammar. You reuse individual components and they interact with each other and build upon each other. Then, the other thing aside from ggplot is, and to underscore kind of the whole philosophy of the Tidyverse, I included a link to this Tidyverse Manifesto, which I think is kind of a funny title, but it basically goes over what constitutes like tidy data principles. So that's like the whole underpinning of the Tidyverse, just trying to organize things a little better. And again, there's pros and cons and people who like it, people who don't. And then the other important thing is piping. So if you're familiar with like Bash and piping there, it's pretty similar. You just kind of, the outcome of a function can be piped into another function, allowing you to just write everything in one line, and it flows from left to right. So for instance, f of x is the same as piping x into the function f. So if that makes sense to you, you'll be ready for this kind of tool. And there's different kinds of pipes. There's some that do an operation in place and instantly update the original data frame. And there's also others that don't care so much about the output, but can share the arguments of whatever you piped into it with the next thing. So there's a few ways of piping things, and I encourage you to explore that a little bit, but this is the very basic one that we'll be using the most. And I think that should be it. So I'll jump into the tutorial. So for this, the only materials that you need right now are some knowledge of base R, which I hope that you've been exposed to. And then I will be doing this in RStudio and R Markdown. And there's many cheat sheets in the website for RStudio. I keep a couple printed out and like pinned to my wall. they're really great, and I encourage them whenever possible. And I'll be using some kind of toy data sets. I bring in some external ones and then use one that's included in R. So the ones from the US Census Bureau and the USPS are just 2 tables that I got from the internet, and I will share the files for that. And then of course, any new package installs, at the end of the tutorial, I include my session information so that you can see the versions of everything I'm using in case that you want to recreate anything and are having trouble. So this is the table of contents for the tutorial. It looks a little crazy, but basically I want to do 2 things: just introduce you to using the very basic functions of the Tidyverse, and then doing a bit of stats with tidy models and some other Tidyverse extensions. So I'll be using mostly Tidyverse packages, but I'll also like pepper in some of my, some of the packages that I find useful from other kind of sources that play well with Tidyverse. So I think I will switch gears to the tutorial. And I think for just for the sake of going over everything, if we could save questions until the end, I will be happy to stay after and talk to people. Okay, so is that sort of visible to everybody? A little bigger? Sure. Is that decent? Okay, so I really enjoy using RStudio. I think everything's really easy to see. This is where your variables show up. This is where my code is, and I have a console where I can try one-off things that I don't want to code into the future. And then there's all these really helpful tabs for when you want to plot or you want to find the manuals for functions. So if you're not familiar with RStudio, I highly recommend it. And this document is an R Markdown document, which has the advantage that you can do some text formatting on top of including code chunks. And in the end, you can knit it to an HTML or PDF, and it's super useful. So without further ado, I will start just running everything. So the first thing that I'm going to do is just load my libraries. Tidyverse is obviously the first one, and then tidy models, and then one plotting package that is called ggRepel. That's really nice for plotting. So the first thing is just reading the different tables that I'm going to use. So the first one is state areas and state abbreviations. So I will read them really quickly, and this is just using read TSV. And again, so the typing here, I'm choosing not to type my columns, but that's really up to your preference, but we're doing really simple things here, so I don't think it's necessary. So then number 1, using the Tidyverse to wrangle and display the data. So the first thing that you want to do is just check that everything has been read correctly. So this is state data, so around 50 rows seems like it would be right. So that's kind of the first sanity check. And we're familiar with functions like head, which give you a peek at what your tables actually contain. So for state abbreviations, we have every single state, and then we have the abbreviations that they use in different years. So here's the first use of piping. So instead of using just head, I will take state areas, the data frame, pipe it into a range, which will arrange it according to some column. And in this case, I want reverse alphabetical order. So I even put this descending argument. And then I lastly pipe it into head. So this is the same kind of outcome if you had used just the nested function calls. So then the second table that I have on top of the abbreviations for the states is the name of the state with area data for total state area, as well as total land and total water. So this is kind of, again, not a super biological data set. It's just kind of a toy data set to start to manipulate things and see how we would deal with some problems. So there's a couple of functions where you can, so for these small tables, using head or things like that, it's really fine because they're so small. But if you're using your actual work and you have thousands of entries or hundreds of thousands of entries, it's not so great to be manually scrolling through like so many rows. So some things that you can do is use functions like glimpse, which gives you a peek at what the types are and how many. And basically just includes everything that I did manually up until now in one function call. So here you get the shape and the types and everything. There are a couple more functions to look at your data frames that I will cover in just a second. So we have these two tables. We want to start asking questions about them. So the first thing that I'm going to do is come up with a made up category. We have state areas for land and water. So I'm going to come up with a category that's just called, is this a dry or a water state? And it's purely based on the percentage of area that is land versus water. So this is just a made up category. We'll see if it's useful. So the first thing that I'm doing is I'm making a new data frame, and I am piping state areas into this function called filter, which does what it's advertised to do, which is filtering. So first thing that I want to do is take out the United States, because if you notice, well, actually you can't see it here, but there is an entry. Oh, there it is. There is an entry for United States. And as we know, that's not a state, that's the whole country. So, and then I will take out the District of Columbia, apologies, but that is a city more so than a state. So it would be weird. And so the other thing that I want to do after filtering is I'm piping the outcome of that filtering into select, which again selects columns. So I am only keeping name, and to avoid redundancy, I don't even want to name the columns that I'm keeping. I'm just telling this function, just keep whatever ends with mi, so keep whatever is in miles. So you can do a lot of things, not just based on the name of the column, but also on the content of columns. You can say, oh, just keep the ones that is car or is double. So you can only keep the numeric ones or the categorical ones. So you don't even have to know like what your columns are named. And that's also helpful when you have like funky names of columns and you just want to subset by data type. And then lastly, so after filtering and selecting the columns that I'm interested in, I finally create this category of % water by dividing the columns that I know about. And then category, I just did a quick if else statement. So if the water % is more than 20, then it's a water state. And if less than 20%, it's a dry state. So this is kind of the just something that we're going to come up with and see if it's actually useful. And so the other thing that you can do is rename your columns. So in this case, state abbreviations had these bunch of years as column names, and we don't like numbers so much as column names. And so the next thing is to, oh, and the other thing is that we have a bunch of state abbreviations from past years that are not really super useful to us, like Arkansas is RT, and I don't even know what that means in 1831. So we just want to keep whatever column ends with present. And so that's what we do here. We select name as well as whatever column ends with present. And then we rename whatever column ends with present. Again, we don't even want to type that column in to abbreviation. And lastly, the mutate function is the same thing we did here. So just create or modify like an existing column. And what I'm making sure of here is that using, my string manipulation, that each abbreviation is only two letters. So then this is just taking you through basically cleaning and joining tables. And so the next thing that we want to do is join the two tables by name. And you will often have to specify what column you're joining things by. And this is another case of kind of where tables might come in useful. Like if you have different types of the column, even if they have the same name, R might not like that. Sometimes, you want to, sometimes you have things that are not compatible. So that's why you might be interested in keeping a more rigid data structure. So here we're just joining by name, which we already know will work. And so it's great. We have all our data in one place. So then the other way of looking at your data kind of in a big picture is this cool package called NANIR. I'm not really sure how to pronounce it, but this visualizes whatever you're missing in your data. So I think this is really great for seeing in your columns what % actually made it through. And here we have a pretty good scenario, but we do have some missing data. I see some chat. Oh, okay, great. Yeah, so I will talk about that at the end, but yeah, if you see any repetitiveness here, it might not be necessarily because you have to do it. It might be for clarity of writing. So definitely take note, and I'm happy to talk about it more. But in any case, so this function vismiss, I think is really great because then you can see like how sparse your data is and how incomplete it might be. So that's really helpful. And another one that I really like is skim, which comes from this other package called skimmer. And what I really like about it is that not only does it tell you the types of your columns, it also gives you tiny histograms at the end so that you can see what's going on. So basically, our join went well, but we want to exclude the columns that did not have a matching abbreviation. So that's just quickly something that we can do with filter. So filter out anything, or sorry, filter and keep anything that is not NA. So here's the negation. So I'm going to run that, and yeah, so our states are still there. And so that's basically just looking at your data, seeing if everything's going correctly, cleaning it up. And so you want to start asking some actual questions about this data. So when I was putting this together, I was kind of wondering, what can I do with just like land and water? It turns out not a whole ton. So I will bring in like another data set to actually make it a little more interesting. But first, we just want to kind of get some summaries. So I have a suspicion that my percent water might not be a super useful metric, and it might just be reflecting states with a really tiny area that get an overblown percentage of water rather than any conspiracy to keep all the water in like a certain part of the country. So I want to summarize my data, and I want to group it by category. So category was that whole like water state, dry state that I made up at the beginning. And then what this does is that for every group, aka for water states and dry states, it calculates whatever you wanted to in this summarize function. So here, I just wanted to calculate the mean total area. So to see all the tiny states ending up with a high percentage of water just by virtue of having a small area. And I will display that. And yeah, actually the water states seem to be pretty tiny compared to the dry states. So there might be some confounding here, and it's not really super useful to look at. And so another thing that we can do is change the shapes of our data frames with pivot longer and pivot wider, which if you learned the Tidyverse before, they used to be called gather and spread. So I think actually after having to learn pivot longer and pivot wider for this tutorial, I think I prefer them to gather and spread. I think the arguments are a little more intuitive, and hopefully if you're familiar with them, you'll see why. So basically, I usually just have really repetitive variable names. I'm just saying, this is the same data frame, but longer. So states percent water is going to be pivoted longer. So I'm grabbing the columns that end with miles, and from those column names, I am changing them to area type, and then whatever is in the columns, the values will be changed to area square miles. So hopefully you can see what's actually happening here. So whereas before we had one entry per state, now it basically has some repetition for the categories that we're not so interested in, because it took the columns that were named square miles and land square miles and made them kind of basically, instead of having the wide setup, it made them longer. So there's some repetition, but at the same time, it might make it easier to plot some things or calculate some things, because now you can do that whole summarize and group by, and you can do that by area type. Whereas before, when they were individual columns, you could not do certain things with them. So reshaping can be really useful sometimes, depends on what you're trying to do with your data. So this was pivot longer. And now I think we're ready to visualize some of what this looks like. So the first thing is just a simple histogram of the made up metric percent water. And that's pretty easy. It's just ggplot, you give it your data, and then you add this geom, which is a geom histogram. So the geoms are basically all the layers that I mentioned before. So in this grammar of graphics, you can basically layer geoms on top of each other, and they will interact according to the specifications that you give them. So one of the key things here is that when you do this ggplot call, if you give it the data here, this data will be inherited in every subsequent layer. And so you want to make sure that if you want your layers to share the data that they're drawing from, you want to either put it here, because if you put it here in geom histogram and you add something else, it's not going to work. And then the second thing, aside from the data, is those aesthetic mappings that I mentioned at the beginning with the textbook. So your aesthetics are just things about the appearance of the graph, and then you want to link it to some columns in your data. So here I'm telling it that whatever gets plotted onto the x-axis is coming from percent water. So aesthetics can include size, can include the filling of your dots, of your charts, can include many, many things, and they're common to a lot of these tidyverse functions. So that's a nice thing. Like once you figure that out, you can really share that across the board. So that's a very simple histogram. So now, what if we want to do, what if we want to just differentiate this by the water states versus dry states? Well, we can use this really useful function called facet wrap, in which, again, I mentioned the now before, you can break things down by area type. So, so you can subdivide this histogram, and that's going to look something like this. So basically breaks it down into 3 different histograms. And then another cool geom is geom density, which is basically the same information as a histogram, but kind of normalized. And another thing that I'm doing here is that I'm saying scales are free, because if you notice, if you notice, all these scales are slightly different. So if you're interested in the overall composition, scales free might be a good option for some of these plots. And so here's another sampling of things that you can do with ggplot. So here, the first one is geom violin, and then I'm choosing to do, again, because of the scaling thing, I am choosing to do scale y log 10. And so here we can see, okay, great. Definitely our dry states tend to have higher land and total areas, and then our water states, again, not to beat a dead horse, but it's still a confounder for tiny areas. And then here's just an example of something crazy that you can do with ggplot. If you just keep plotting things on top of each other, you might get something really ugly, but it's technically correct. So you would just be careful with that. And then obviously your bar charts. So these are respectively geom violin, geom jitter for a slightly more messy version of geom point, and then geom bar, which you can stack or fill depending on what you want. So lastly, I want to show you how to customize plots a little bit. So instead of just calling ggplot, I'm saving my plot onto this object called sp for simple plot. And that's a simple scatter plot. So this is just, again, our data, and then our mappings to geom point. And then if I want to scale everything by maybe some custom metric, I can just make some function and then mutate my data frame to add like a custom scaling for dot size. So that's if you're just trying to customize this a lot. And the reason that I would customize this is because if I just say dot size equals area, we're going to have really huge dots and really tiny dots. So I wanted to do something custom. And so this is just a function that I made where basically I get the closest quantile to my state area. So I divide all state areas into steps of 10%. And then I find the minimum of the difference of the absolute difference between the quantiles in my state area. And then the index of that quantile divided by 10 will be a numeral, being like, oh, if it's 0.5, it's closest to the 5th quantile or something like that. So then once I do this, I can have different dot sizes, and that's great, but we still don't know what states these actually correspond to. But interestingly, all these wet states are over here, and then all these dry states are over here. So then, yeah, so not too much to it, just another mutate and another ggplot with geom point. And then the labels, which is the captions that we want and the title that we want. And we see them here. They're still not very fancy, but we can still customize it even more. So one of the things that I like to do for my plots is I create like custom themes for myself. And usually I just name them something random, like theme minimal, theme 0, theme 1. And I build them on top of classic or some other blank theme that comes with the default art. And then if you add this theme function, you can basically customize just everything you want, like the size of the fonts, the face of the fonts, etc. So once I do that, I can also customize the scales. So I don't like so much these red and blues that art does by itself. So I want it to be firebrick red and deep sky blue instead. And another thing that I want is I want to hide that size legend, but because it doesn't really, the exact value of the dot size is not really important to us. And then another thing is that I want to add this theme. So then that's my new customization. And then the last bit that I want is I want to add geom text, which will actually put the labels of the states and finally use our abbreviations in order to get an idea of what's actually happening. So then it's a lot of lines, but they're all really obvious. And then in the end, we can have some plot that looks a little bit nicer, and you won't be like super ashamed to present it to your PI. So that's the point of this, to sort of make something a little more cleaner. And so once you have that, some of the issues that we can have sometimes is a bit of crowding with the labels. Here in the data that I'm using, if you notice, I'm feeding it a different data than what I gave it initially, because I'm saying for the geome text, I don't want you to take the original data. It's going to be too crowded. So what I did was I filtered the data. And so even though the underlying layers are using the entirety of the data, the text is only using that subset that is not going to crowd my labels down here. So I hope, that's kind of a hack, but another alternative to doing it that hacky way is that you can use this package called ggRepel, which instead like automatically repels your label names from your points, and you can sort of adjust like what the force of the strings are and like how far away they can be pulled. So it's kind of neat, and solves that crowding issue sometimes. And so, okay, I rushed through that, but hopefully most of it was familiar to some people and easy to understand, and nothing too crazy. But I think we have 15 minutes left. So I think we can definitely take a couple of questions now, if there's some. Yeah. Oh, sure. So the question for those on Zoom was going over the mutate function. And then let me see in the, okay, and why did I have to do a second ends with if I had already selected? Okay, so let me go over the mutate function and the ends with situation. Yeah, so let's see. Oh, okay, so I'll address the ends with because it's right here. So here I'm doing two sets of ends with, and the whole idea of this is that I don't quite know what my data, what my columns are named, but I just know it ends with present. So here in this case, I'm selecting name and ends with present. And then I have to specify again what I'm renaming, because I want to just rename the column that ends with present to abbreviation. Like I don't want to touch name. So, if you can also just give it the actual column name, but for this, like you do have to specify the column even if you have selected. That being said, like you can just select at the end, it doesn't really matter. Yeah, I think the selecting is just for kind of keeping things clean and keeping things small, but I hope that made sense. Like you just have to specify which column, and ends with present is again like just doing the same Boolean operation that you select with, but you can do them in any order to achieve what you want. And then the other thing about mutate, so you basically have all these operations that have been going on before, and you're piping them into each other. So then when you pipe them into mutate, this is the new column that you're creating with mutate. And mutate, you can create a new column, or you can mutate an existing one. So in this case, I'm creating a new one, and then the nice thing is that you can just include the names of your old columns or the ones that are already piped in, and then just make something new. So it's new, and then equal sign whatever operation, whatever mutation you're doing. And then you can keep including columns here and like doing extra things. So you can have a whole like mutate set that you're adding. Is that a little more clear? Okay, great. Any other questions about this? Yeah.
Yeah, the filter and select functions we were just going over.
Yeah.
So is the difference that filter is always working by column and select is working within values?
I think you may have gotten it backwards. So filter is filtering based on values, and then select is based on column names or column kind of attributes like, oh, is the character or is the double? Did I understand right?
Yeah.
Oh, sorry. And for the people on Zoom, the question was filter and select, which one works with kind of column names versus column values. Yeah. So does that,
Always the case that
I'm trying to think. I think so. So filter will filter like based on, some Boolean that you set up, like, oh, just filter whatever equals like this value, or filter whatever equals this string. So it will remove rows based on what you say, right? Whereas select, it will remove columns based on their names or like their values. So I think that's like the key difference. The filter will remove entries.
Yeah, so it's not that select just only performs the usually function and kind of leaves everything else there, but it also removes data that doesn't comply with.
Yeah. For select, you can say like select name and like, water content or whatever. Or you can also say select name, water content. Actually, sorry. You can basically do like a positive selection and like keep the ones that you want, or you can put a little minus sign and then it'll knock out one if you just want to get rid of one and keep everything else.
Yeah.
So you can do both with select. Yeah, so filter, again, like it's more filtering out entries, whereas select, it's like cutting down your data frame to select columns. Yeah, thanks for asking questions. Yeah, and I'll send out this RMD file for people to to explore a little bit and rerun if interested. But yeah, this will all be sent out at the end of the talk. Okay, great. Cool. So in the last 10 minutes, I will quickly run through just, again, a sampling of what you can do with tidy models and some of the more pointed questions that you can be asking of your data. So this is the whole plot customization part. And so the other independent data set that we're bringing in to make things a little bit more interesting is this default data set from R, which is called US rent income. And you can find all the data that R comes with if you just type data in parentheses. So again, I'm using skim here because I like the small histograms. And basically the information that you have here is a GeoID, which is just a state number. So from 1 to 50, I think. The name of the state, and I think, actually, yeah, I'm not sure what this variable thing is, but the estimate and MOE. So if you look at it, yeah, there we go. So the variable is income and then rent. And then the estimate is like the estimate for each of them. So here you can see already, we might want to do some reshaping based on that. So that's the data summary. And then we want to know, is rent and income in any way related to water content of the state? Again, a totally weird question to ask, but just suspend your disbelief for the duration of this talk. So then we have, again, some transformations that I want to do. I don't want this name in all caps, so I'm going to do a rename. So again, rename is new equals old. So this is the old name that I want to get rid of. Then I want to select. So this is what I was telling you. I want to just get rid of this MOE because I think that's an error measurement, and I want to keep things simple. So and then I want to remove this GeoID because it's just a number from 1 to 50. It doesn't tell us anything. And so, we're doing this renaming, this selecting out the columns that we don't want. And then the next thing is I want to pivot wider because that whole redundancy of having 2 entries in each state, I don't like it so much for matrices and later analysis that we might do. So I'm pulling names from variable and then values from estimate. And that'll allow me to have everything in one column. So here's what that will look like. And so whereas before we had, that variable that had income, rent, etc., now we have them into separate columns. And then lastly, I just like to throw this in sometimes and I omit in case that there's some missing data. Oh yeah, because the last thing that I did was a left join. And so this NA omit basically omits any missing data that did not have an entry in each of the tables that I joined. So again, whether that's great practice or not depends on the data that you're dealing with. Don't throw out anything that you might want later. And so now we have all state data, which is income and rent, and then our information about land. And so one cool library that is not Tidyverse that I also like is ggally. And this literally just do ggpairs and call one function, and you can have this this really great overview plot of all the correlations between your variables. So if you want to know like what variables are actually orthogonal to each other, this might be super useful because you can clearly see. So here, not surprising, you can see that rent and income are really highly correlated to each other. It's more expensive to live where people get paid more, etc. So that's unsurprising. And then another unsurprising thing is that total square miles, kind of unrelated to income, kind of unrelated to rent. Land square miles, again, nothing really going on here. Water, again, nothing really going on here. So rent and income seem to be pretty unrelated from our water data set, which is like, you know, we saw that coming. But then percent water is doing something kind of weird. It's like really related to rent, I think, but then it's not really well correlated to land. So that's a little bit strange. So we'll keep investigating, we'll keep beating this dead horse of the percent water. And so another cool library, I think this one is actually from Tidy Models Core, is basically doing the same thing. So the correlation between all your variables, but it returns a nice data frame and it does slightly different things. So here, the first thing that I did was I grabbed all numerical state data, which is what I fed into GG pairs. And sorry, I didn't go over that. I think I rushed past it, but the first thing that I'm doing for all state data is that I'm taking out dot size. So that's a negative kind of select. And then I'm only selecting if a column is double. So I'm only keeping the numerical columns because I just want to do a correlation instead of like including my categorical variables in some sort of other test. So I just want to do a normal kind of correlation. And so using that same numerical state data, I did the same thing. So piping all the numerical state data into correlate, and then I rearrange it by the actual value of the correlations. And this function shave shaves off the upper half of the triangle because it's just the same as the lower half. So as you can see, it gives you some really nice data frames to work with. And there's this text display in a nice format using fashion. And then our plot will actually give you this cool plot of the direction and sort of magnitude of the significant correlations. And as you can see, water area is correlated to itself, and then income and rent. Actually, income and rent don't seem to be super correlated. But another cool correlation plot that's kind of fancy is a network plot. And here you can clearly see that the water and land variables are really correlated to each other, and rent and income are correlated to each other, and percent water is off doing something that nobody knows what it is. So that's some fancy correlation plots that you can do of your variables. So what I'm seeing here is that we have two mostly orthogonal like dimensions to this data set. So what that tells me is I can get at least two principal components out of this. So naturally, the next thing that I'm going to do is PCA. So if you're familiar with PCA, then this is great. But if you're not, maybe this is not the best way to learn about PCA, because again, you just call one function and you get some result that you maybe don't understand. So that's my caveat with all of this. It's obscuring a lot more steps, but it can be quite simple if you're just trying to get things done. So this is just my little summary. Unsurprisingly, you know, our data sets are not super correlated, and percent water seems to be behaving weirdly. And so we can use this to do PCA, and then we expect PC1 and 2 to reflect these two orthogonal data sets. So then the first thing that I'm going to do is make this into a matrix. Make this into a data frame so I can change the row names and I can keep the states sort of for labeling later. And then running PCA is just principal component, the matrix that you have that is just numerical, and then center and scale, which I won't go into too much detail of. But then the next thing that so this literally just runs PCA, and then you have everything that you need. So from PCA dev, this is where I get the standard deviation explained by each principal component. So I'm making a data frame with that, so this is just PCs 1 through 6, because we have 6 variables, and then we have that standard deviation. So, and then we have the variance, which is just standard deviation squared. And then we do a cumulative sum of the percentage of variance over total variance, so that we can see how our principal components start explaining the variance in our data set. And then lastly, we just throw this all onto ggplot and get some idea of how our principal components are explaining everything. And what I'm doing in ggplot here is, again, the first thing that I do is geom line so that I can connect my points, and then put a point on top of that and do some kind of customization with colors and fillings. And so this is what that looks like. And again, I'm throwing on this clean theme that I made a while ago, just so that you can see that it works for everything that you might want to plot in ggplot or any tidyverse. So again, some really clean plots. And so I have these principal components now, and I want to see what is going on. And we had also predicted that this % water wasn't going to be really explanatory because it was doing its own weird thing and not really correlated to the other variables. And we can check that based on the PCA loadings that go into our variables. So then here's me using ggRepel again and the actual PCA values. So unsurprisingly, here you see Hawaii, Maryland, Massachusetts, Rhode Island, Delaware, etc. So small kind of coastal states are grouping together, which makes sense. And then we have bigger, more inland states up here. And then we have Alaska, which is a giant coastal state. So that's the outlier. So this gives us an idea of what's going on in the data. And then another library that I like is Pretty Heat Map, or pHeatmap. And that's just a quick way of making heat maps of your data. You can cluster the rows and columns, and it's really useful for gene expression things. So I like to use this package a lot. And it has a ton of more arguments that you can sort of customize. So then, as we predicted, total square miles, land square miles, and water, that looks like it has the highest loadings for PC1. And then also pretty high loadings, just in the negative kind of direction. So we care about absolute loadings for income and rent over here, and then % water for some reason, but that's PC2. And then the rest of the PCs are associated with one or the other a little bit more. And so the very, very last thing that I'm going to run through really quickly, just so that you can see how easy it is to see. So now I have my PCA, and I have a bit of noise reduction. I have all these variables boiled down into 2 principal components. The next thing that I want to do is maybe cluster this data. So I'm just going to do a simple Kmeans clustering and see what that looks like. And so Kmeans clustering, this code was slightly adapted from the Tidy Models tutorial page, which has so many more materials. Tidy Models can do a ton more things, but again, that would be another talk. So then the quick thing that I'm doing here is, again, a select of just the first two principal components, and then literally just doing K-means clustering and a summary. So this is just, I think, base R, nothing exciting here, 2 centers, but we don't really get a glimpse of too many differences between states because we just have 2 clusters. So this is just, again, kind of labeling things a little bit more. And I realize that I'm running out of time, so I'll just quickly show you the last two. And again, this is just the clusters that every single state was sorted into, not super informative. So the other thing that I do here is that I just iterate over case 1 through 6, and I do a K-means clustering for each value of K. And I grab basically this augmented data frame, which contains the labels. And it has a tibble, which is another version of the data frame for each single K. And then the very last thing is that I plot every single iteration of K that I did. And if you notice here, I think this might be the best one because it has, or actually, well, you can pick whichever one is your favorite, but the previous time that I did this, I think it changed this time around. Alaska was off on its own kind of cluster, and then different clusters were here. But yeah, this shows you how the clusters change with each K iteration. And it's really, really a neat package and can be really quick to learn. At least it was really quick for me to learn it. And so that's everything for the tutorial. I hope I gave you a good glimpse of all the different things that you can do, from simply transforming and exploring your data to doing a bit more complex correlation in PCA and just visualizing everything you got going on. So I will stay on happily for any other questions, but thanks so much for your attention, and thank you guys for your help setting up and organizing this. Yeah, thanks.
Monica?
Yeah, thank you.
Yeah, we have this room, I think, for a little bit longer. So if people have remaining questions, we could answer them here. Else, I think we'll be sharing all these resources online on the website. So you can go back to that. And,
yeah, thanks.